Okay, good evening, everyone. First, I'd like to thank you all for coming out on this dreadful day. I know it's a little bit better now than it was this afternoon, but it's quite the weather out there, so congratulations on getting here in one piece. My name is Jane Pickering. I'm the Executive Director of the Harvard Museums of Science and Culture, and I would like to welcome you to this, which is the third in our Evolution Matters series for the Harvard Museum of Natural History. And I'm delighted to welcome our speaker who has, moved, Dr. Zhe-Xi Luo, who is speaking to us today about Jurassic grandmothers from China. Before I get to Dr. Luo, I would like to just mention that our next lecture is 2 weeks from now. Robert Hazen of the Carnegie Geophysical Laboratory is going to talk about the new evidence about complex evolving systems driving phenomena beyond life sciences, and such as the diversification of minerals on Earth, and how this impacts the arguments of the anti-science proponents. And this will be on Tuesday, March 12th, here in this hall. I'd also like to I know we have some special guests in the audience, some local teachers who are coming and have a seminar after the lecture, and just to let you guys know that the seminar room is sort of pretty much exactly behind me, right next to the bathroom. So if you go out and just keep going around, then you'll find your seminar room. All the museum's classes and exhibits can be found in this brochure, which is on the table. And so please, if you don't have one, feel free to pick one up. And if you don't like paper, then sign up your email address. So then you'll get everything in your email box about the information about all our programs. And there's also information there if you would like to become a member of the museum and help support our exhibits and our public programs. So speaking of support, I'd like to acknowledge Dr. Herman and Dr. Joan Suit, who are sitting over there, who supported Evolution, the Evolution Matters series. And and they have been dedicated sponsors, thank you, for the last several years. And thanks to their support, we are able to videotape these lectures, and they are available online. So we just put up the first of this year's series by Dr. Arkut Abzamov, who did the first, those of you who were here remember, which was fantastic, on dinosaur to bird evolution, and that's now available, but there are many, many others available, so I encourage you to check that out. So on to tonight's speaker, who's really being welcomed back to Harvard rather than just welcomed to Harvard. Luo is a paleontologist and professor of organismal biology and anatomy at the University of Chicago, and his research specialty is on the evolutionary biology of vertebrates. Tonight, he will focus on the originations of modern mammalian biological adaptations in the Mesozoic era, often dominated by dinosaurs, and story isn't quite as simple as that, and how Jurassic fossils discovered in China shed light on the earliest evolution of placental mammals. He received his Bachelor of Science from Nanjing University of China, and then went on and did his PhD in paleontology at the University of California, Berkeley. He completed his postdoctoral training from 89 to 91 right here at the Museum of Comparative Zoology at Harvard, and then left and spent many years as curator of vertebrate paleontology at the Carnegie Museum of Natural History in Pittsburgh, and in 2004 was named their Associate Director of Research and Collections. He is considered one of the world's top experts in Mesozoic animals, and is counted among the international team of scientists that has discovered many significant species of early fossil mammals, such as the earliest known swimming mammal and the earliest marsupial. He has received a career award from the National Science Foundation, as well as a Humboldt Research Award for senior scientists from the Alexander von Humboldt Foundation. The list goes on, but let's hear about Jurassic grandmothers from China, origins and early evolution of mammals.
Well, my name is Luo, and in English, it coincides with being short. So every time Every time I talk about early mammals in front of my mentor, Fuss Crompton, I'm always a short guy and I shudders. So mammals are very diverse. We have some 40, 50, 400 species. And mammals are also very interesting in that we have some 4,000 genera of fossils. So as a case study of a major clade evolved in the evolutionary history, mammals gave us a great fossil record to work with. Okay? Mammals are also very interesting in the ecomorphological diversity. Let me backstep a little bit. And this is an old exhibition diagram by Willington Gregory in American Museum of Natural History. Okay? And it's a tree. It has many different ecomorphotype maps on top of it. Okay? Foremost, we have all these familiar ground-living terrestrial mammals. Okay? And of course, there are things that can climb the tree. And once you get on the tree, it's not uncommon you can get fully arboreal, and some of the arboreal form could be volant or could be even power flyers. And you can have tongue-feeding termite eaters, and you can have cursorial herbivores, and you can have heavy animals that are graviportal, such as elephants and rhinos, so on and so forth. And then you can have carnivore scavengers, you can have carnivore predators, and then finally you have fully aquatic mammals. Okay? And these are called ecomorphotypes. What the classic natural historians have set up as orders of mammals largely goes with these ecomorphotypes. But of course, the modern molecular phylogeny have already validated from the genetics perspective most of these orders except what we call the insectivores. So mammals are interesting in ecology and in their general morphology. But all this diverse ecomorphotype shares some basic features in reproduction. Okay? And we can, the first group is called monotremes, and these are mammals. They have mammary glands, and they nurse their young, but their eggs were, their young were born with leathery eggs. And in contrast to the egg-laying mammals, we have livebearers mammals. The first group of these are marsupials. Marsupials have very short gestation, but relatively long nursing in the pouch. And here is a figurative metaphor of that. And the most diverse group within the livebearers mammal are placentals. Okay, there are some, depending on how you read the latest phylogenetic literature, over 5,000 species. Generally speaking, the placentals have very elaborated, more invasive placenta that embedded in mother's uterus. As a result, they have longer gestation and more effective nurturing in the uterus. But all these mammals, in addition to their basic reproductive ecomorphological patterns, they also have bones, they also have teeth. It is really through these osteological and dental features that we can tie the fossil record with the modern mammalian diversity. Okay, normally this is how we do it. So we have placentals, we have marsupials, and these are united as therians characterized by livebearers. In addition to their mode of reproduction, they also have teeth and the jaws and skeletal features that characterize them. Okay, and these therian mammals are grouped together with monotreme, all together characterized by mammary gland, and also the derived mammalian bone feature, so on and so forth. Suppose we find a fossil without this soft tissue. It is really these osteological features help us to place them on the overall mammalian phylogenetic tree or family tree. Say, for example, if we find a fossil called Juramaya, or mother from Jurassic, and its osteology and teeth places closer to placental than to marsupial, but not so advanced yet to be inside of the placental, we will call them eutherians, right? And if we find another fossil, say like Sinodelphis, and by teeth and the bones were able to associate it closer to modern marsupials than modern placentals, we would place them close to the marsupial line and call them metatherians. It is really these early fossils that gave us a time estimate about how a particular evolutionary lineage first appeared in the fossil record. But more important than that, and very interesting to me, is these fossils would help to provide what our ancestor would be like if these are closely related to us. And it is really from this ancestral feature from the early fossils we managed to establish the evolutionary pattern, how the modern group arise in the fossil record. Well, now this fossils associated with individual lineage gave us the minimal age of the group, but all this extant mammals would also carry with them the molecular sequences that are part of their history, and through those molecular sequences, we can manage to provide a molecular time estimate. Usually, there is a gap between the minimal fossil age and the rise of, or estimated divergence time of a modern group represented by molecules, okay? But the biggest early mammal diversity are not the group associated with placentals or not the group associated with marsupials. They are associated here in between the therian mammals and monotremes. So these are modern metatherian and eutherian group. There is a large number of groups, okay? And there are some 15 different groups in between therians and the monotremes, okay? And these groups, from my perspective, are the most interesting mammals, and many of them are from Jurassic. And the modern mammal group, in return, is nested inside a group called Mammalia forms, okay? And these are fossilized mammals from very early age, and their osteological and dental features definitely associated them with the modern groups, but they are not quite there yet, okay? So we call them Mammalia forms. And Mammalia form, in return, are descendants from a large cluster of extinct reptilian fossil known as cynodonts. Normally, we refer them as mammal-like reptiles, okay? And of course, we will have this osteological feature to construct this phylogeny. And in the more recent months, a much larger version of this study is published by a team funded by National Science Foundation Tree of Life Mammal Project. Okay, but if you take this genealogical tree and place it on a time scale, you will see some very interesting sequences. Okay, and the next diagram I'll follow from here to here. Okay, but vertically. Okay, we start with cynodonts that are very common in Triassic, and this is even before the rise of the dinosaurs. Right around the time when dinosaurs start to appear in the fossil record, in this time roughly from 220 to 190 million years, we start to have this fossil groups comes into the fossil record, and they definitely have some parts of the modern diagnostic mammalian features. That's why we call them Mammalia forms. And if we move a little more recent to the Middle Jurassic, from 180 to 165 million, we start to see the fossils that we can definitively place on modern clades. That is either associated with modern monitoring to some extent or associated with modern theorians to the other, okay? Well, by the time we get to late Jurassic, up to 145 million, we start to have groups that we can literally identify with eutherians, such as Jurassic. Well, Cretaceous is also very interesting, but if you go from present day back step 220 million years, the first 1/3 of that 220 million years of mammalian history are in Jurassic, okay? And by the time we get to Cretaceous, the game is almost over. So thus, our theme about grandmothers from the Jurassic today. The fossils from this earliest episode really can help us to frame some questions and address how modern mammalian biology really arose in the fossil history. And this is the work that I started with first, okay? So I'm particularly happy that FUS is able to come and probably offer me some criticism after this, okay? Well, the biggest question is, what makes a mammal, right? Mammals have hairs, mammals have mammary glands, sweat glands. These are all integumentary structure associated with the skin, okay? Mammals are also characterized by this very important feature of lactation. Of course, in the fossils, we cannot get the fossilized mammary glands, but because lactation has such a fundamental impact on how mammals grow, and we have related growth and dental features that allow us to extrapolate when lactation really originated. And of course, you know, it's no-brainer, the brains are important for us, right? Without capacity to think, we wouldn't be here to sit and enjoy this discussion, okay? Well, let's look at the hairs and the integuments. And I will start with one fossil, and the name is not important. Let's call it Docodon. It's some extinct mammals from a long time ago. And this particular Docodon has very interesting scales associated with a big fat tail flattened, okay? And in between those tails, you can clearly recognize streaks impression that are the hairs, okay? And in addition to this hairy, scaly tail, and you can also tell that the tail vertebrae themselves are flattened, and they have this so-called transverse processes. Okay, these are some bones sticking out to the side. And if you compare the morphology, they are really very similar to modern beavers and modern otters, and both of which are aquatic, okay? And it is with this kind of feature we're able to understand that this guy definitely flapped his tail like in a beaver-like swimming motion, and has very powerful forelimbs that's good for both digging and also for swimming, such as the case of modern platypus, which is also semi-aquatic. And then its teeth have this recurved, sharp cusp, which is typically interpreted and associated with fish feeding. So, besides the fact that we got a cute beaver-like mammal, the important fact is really we have this very ancient extinct group close to mammals, but not quite to modern mammal, within the modern mammal group, already fossilized with fur. Basically, the mammalian integumentary structure originated well before the modern mammals. And there are additional fossils to give us the same information. Well, you may say, well, it's not a big surprise. Mammals have furs. Mammals have this skin-related derived features. Yes, it's not a big surprise, but nonetheless reassuring that we can have fossil record good enough to give us an understanding of these common sense features. Okay. Next, I'm going to talk about lactation and the growth pattern. Okay. I will start with a fossil that's Fuss Crompton's favorite, and this is called Pachygenolus, and this is from the early Jurassic of Southern Africa. But the larger group of this actually has much wider distribution in Argentina and in Southern Africa. The record of this particular group related to Pachygenolus really goes back to late Triassic, as old as 220 million years. Okay? And if you look at the very interesting pattern of this Pachygenolus fossil, you realize that this PC are not politically correct, and they are called post-canine. These are the teeth behind the larger canine. And if you look at the pattern of this, you can definitely, in a single fossil, recognize at least 3 generations of teeth. So this guy have multiple replacement. Besides the fact they replace quite often, multiple times, they also have a alternating replacement. The fact is probably not as important if you compare it to the modern therian mammals, okay? And here is a Australian dingo, okay, and it belongs to placental canis relative to modern dogs, okay? Younger individual, we have deciduous teeth. Older individual, we have this permanent generation of teeth, which are called molars, okay? When these molars, when these deciduous teeth are being replaced, it goes sequentially. Okay, the premolar or relatively simpler teeth behind the canine, when they replace, they go in a sequential way. Okay, from this end to this end, and then there are marsupials, and marsupials has one single tooth being replaced. Well, the question we can ask with this early fossil is how we get from the ancestral condition of our near relative to our kind of condition. The reason we care about this is this pattern is related to determinate scalp growth and the lactation, whereas this is essentially our ancestral condition. Let me backstep a little bit, explain what we call determinate scalp growth, okay? I'm going to digress and use an example from the other side of modern vertebrate diversity, and that's the birds and the dinosaurs, okay? We have dinosaurs have very rapid early growth, okay? And the modern dinosaur like ostrich and the emu, facilitated by this intense parental care, they grow really fast early on. But once they get to a certain stage, they reach adult body size. For rest of the longevity, they have this plateaued growth curve. In contrast, dinosaurs and the birds, there are many reptiles that would have the slow and the sustained growth, and this is what we generally call the indeterminate growth, okay? Let me redefine it a little bit, okay? And this particular diagram is a contrast between a rodent, a representative of modern mammals, and a crocodile, okay? And generally speaking, in non-mammalian, non-avian vertebrates, you have the slow early growth, but the growth never quite stops. Whereas in birds and also in modern mammals, facilitated by, in mammals' case, lactation, you have this rapid burst of early growth, but because you grow so fast, you have to taper off somehow, so you have this plateaued growth curve. And this gave us a very interesting body size difference, and if you have good fossil preservation from some fossil sites, you can literally test, okay? And you can distinguish if some sample of fossils actually better indication of prolonged juvenile growth, and then a long, sustained, but slow growth throughout the life. And if we have the case like this, we would call it indeterminate growth. It's not really completely without end, but the sense is it will go on for quite a while. But if we can do the statistical analysis of a decent fossil sample, we can distinguish a fossil group that has achieved very high rapid growth, and then their growth, once they reach the early age, will be leveled off, okay? Now here is some real data, okay? And this data are some small rodents, small insectivores, compiled by a very active research lab in the 1970s in Smithsonian. John Eisenberger was the main leader of that, okay? And these are the days of individuals that you can sample from the field. And these are 100% of your adult body size, okay? And if you map out the growth pattern over a time course, you get a sense of many of the rodents pretty much matured, reach their adult body size 30 days. Very, very fast. And some of the insectivores will go on a bit longer, and they will go 60 days, 70 days. But the bottom line is all mammals grow very fast. Why they grow fast? We all benefit from mother's milk, and we all benefit from mother's lactation. But once we get up to that adult body size, our body size is fixed, and therefore we have the fixed plateau. All right? And it is really in this early phase because we benefit from the lactation, and we can grow without teeth, and that shaped our basic growth pattern. So everything else being equal, because we have to plateau off, we have early termination of the possibility to replace our teeth. Therefore, this lactation really gave us a different timetable for growth. And if we are able to establish the so-called limited dental replacement, called diphyodont dental replacement, we are able to infer that this particular fossil must have had lactation. So that's my reasoning. And here is a fossil, okay, and it's called Brazilodon. It's from late Triassic of Brazil, okay. And this is a young individual, this is the older individual, that's the time course. And you can tell that the front part of the teeth is still reptilian, okay. Whereas the back side of the teeth, and they are having this sequential replacement, there are no longer this rapid replacement of teeth, therefore more mammal-like, if not yet fully mammal-like. Now, if we go on in the evolutionary tree to another more derived form called Cynoconodon, and this is one of those mothers from Jurassic, in early Jurassic of China, okay. This is actually a work that I did with Fuss together, okay, and this is a very young individual. This is a very old individual. I'm not going to quiz you about this very complex diagram, but suffice to focus on this anterior part. It's called canine, and even in the youngest individual, we have replacement of canine. If you map it out, you'll find the following pattern, okay? And that is the anterior teeth are replaced multiple times. And we can actually know for sure at least the Cynoconodon has 4 generations of dental replacement. It'll replace early, it'll replace late. Basically, it keeps on replacing, it never fully stops. And that is a reptilian pattern. Now, if you compare this fossil, Cynoconodon, with another fossil that are relatively well known called Morganucodon, you'll find a size pattern is very different between the two species. Essentially, Morganucodon has very narrow growth band, and it fits the prediction of the general model. If you have a sample of a fossil, very few babies, relatively few adults, and they have very narrow size range. In contrast to that, Cynoconodon has a much wider range. And in fact, for the range of Cynoconodon we have, we estimate it can grow almost like 40-fold of the body mass, okay? But the key is the small Cynoconodon replace their teeth, the larger Cynoconodon would replace their teeth, and essentially minus the fact that Cynoconodon looks like a mammal with its jaw, looks like mammal with its ear, but its dental replacement is reptilian. Okay, so what does this tell us? Okay, it basically tells us that in this near mammals, we start to have more mammal-like condition. There is reduced replacement of teeth. And in Cynoconodon, we already have very likely a single generation of most of the teeth in the jaw, but the anterior teeth is still replaced multiple times, early and also late. It is really in Morganucodon, we start to have no replacement of molar, therefore achieve this truly modern mammal-like diphylodont dental replacement that's usually facilitated in extant mammals by lactation. So if you take this logic one step further, essentially between Cynoconodon and Morganucodon, we caught the evolutionary moment when we start to have the earliest fossil that can tell us reliably lactation has a reason. Okay, well, I wonder if we have dental care professionals here. Okay, and you would wonder, every time you go into the dentist's office, they have all this fancy gadget that makes noise. You open your mouth, half sedated, and they do this work in your teeth, and it's suffering, right? Okay. Well, the bottom line is we have limited dental replacement. If we get stuck with that permanent dentition, we have to take care of it, right? Okay. So essentially, next time you tell your dentist, it's really that moment 220 million years ago. Evolution took a turn for the better, so you get to suffer, and he has a job. Well, let's move on to the brain, okay? Okay. We have this very nice fossil, and this is prepared by the very best fossil preparator, Bill Amaral, who worked here in the MCZ for many years, have just recently retired, okay? And besides the fact we can figure out many anatomical details, and this is what we know about this particular fossil, okay? And it has a relatively large brain case, okay? And we name it a hadrocodium. Hadro means full, codium means big head. And so this is also a study that I did with Fuss, okay? Now, if we scale the size of the brain against the estimated body mass, we have this EQ. It's not emotional quotient, okay? It's encephalization quotient. It's a metric that you estimate body size after you correct it for body mass, all right? Okay. And so the EQ is 0.49 in contrast to a modern opossum that has an EQ of 0.34. So this guy is brainy compared to modern opossums. Here is how we get it, okay? And here is a CT scan surface model, and in the inside we can see quite a bit of anatomical structure with quite a bit of struggle from my co-author Ted McCraney. We're able to extract this brain endocast, and you can see many features, and such as a olfactory bulb and a relatively large cerebral hemisphere, and not just a large cerebral hemisphere. it's really the side of the cerebral hemisphere homologous to the brain cortical area in modern mammals for sensory touch and motor coordination that got significantly bigger, okay? So what does this mean? Well, let's pull back and do some comparison first, okay? On the left, it's our distant relative from early Triassic called Thrinaxodon, okay? It's brain endocast, okay? At first, we can tell that the relatively modern mammal have very distinctive olfactory bulbs, okay? And the biggest difference is modern mammals have this large cerebral hemisphere, okay? And modern mammals also have a cerebellar hemisphere that is absent in the distant cynodonts. But when you add on top of one another, and you can tell that this guy is not very brainy, and this guy is brainier, okay? And so the question is, how do we get from our Triassic ancestor or ancestor-like condition to the modern mammals? Well, here is how we do it, okay? And we use the evolutionary tree as a roadmap, and we essentially map this transitional stage, one state at a time on the tree to see how it distributes. And this distribution pattern will give us a possibility to infer how the process of evolution actually occurs, okay? Now, with the rise of mammalia forms, these are by jaw, by teeth, characterized as very mammal-like. We start to have this divergence of the brain hemisphere, okay? And once we get into modern mammals, and we start to have very distinctive midbrain areas differentiated, it is really in the modern Therian mammals we start to have this cerebellar hemisphere. Let's focus on next, this general anatomical region, okay? In modern mammals, this general area of the brain, the neocortex, is responsible for sensory touch, okay? And you can actually map this somatosensory fields, and so you have this representative animals, okay? And the signal should come from a tactile sense of the hair. We know that the early mammals got hair, so that's not a worry, okay? And here is a diagram. Here is a diagram. I'm sorry this is washed out, but you've probably seen this multiple times. It's a cut-out section of the brain in the cerebral hemisphere, and it represents the sensory touch of the surface, okay? But if you look at this Morganucodon, okay, you get a sense that the brain cast become larger, not just become overall large. It's really in this limited cortical area that in modern mammal responsible for somatory sensory fields that becomes larger. So what this means is in the early evolution of mammals, we start to have large olfactory bulbs, large cerebral cortical area responsible for tactile sense, okay? And this is before we get to the modern mammal range of a brain size. Again, this EQ is relatively brain mass after being corrected against the body size. And in hydrocodone, we start to have the relative brain size really within the range of modern mammal variation, okay? And after that, it's already, the story is largely over about the brain evolution, okay? So we talked about what we can do with the fossil from the Triassic and the Jurassic. Well, let's forward relatively rapidly to talk about the split of modern groups, okay? Modern groups of placentals and marsupials, all right? Here is the fossil. We call it Juramaya. It's 160 million years. And I'm happy to report that we now already have a second fossil of this that we are working on, okay? But before I go into that, let me go back to our breakfast egg. And that is, here is the embryo, and the yolk, yellow part is the yolk sac. Much of it is allantoid sac, and here is the amniotic fluid sac. Okay, essentially, to live within the eggshell comfortably, we have to bring our grocery with us. Okay, we have to have a nice hot bath, and then not enough, we carry our poor potty with us inside the eggshell so we can live very comfortably. Now, to take those homologous parts to understand the placenta of modern mammals, marsupials largely have a yolk sac placenta. Okay, so this is a general feature of marsupial placenta, and this placenta is not terribly integrated into the endometrium of the uterus. And then placental mammals also got a placenta, but it's more sophisticated. It's from the amniotic part of the extraembryonic membrane, and this placenta structure deeply embedded into the uterus. Therefore, we have much more effective parental care in uterine. Now, if we look at the broader pictures of placental diversity, no matter which group you are looking at, they're all having largely the same type of placenta. Okay, marsupials are different. Okay, and the parental care is much more limited within the placenta, and usually marsupial also have much shorter pregnancy. Okay, and after the precocial fetus were born, they are sucked onto the mother's nipple inside the pouch, where they have the rest of the development. All the marsupials tend to have the same general placental design, except the bandicots, which are more like the modern placentals. So there is always exception. But all these features are also associated with teeth, with jaws, and it's by really looking at the teeth and the jaws, we're able to map some of the earliest fossil onto the overall placental and the marsupial trees. Okay, this also matters from another perspective, and that is we now understand mapping the genome or the complete genetic makeup is very important. Okay, and so since 2000, after 2005, multiple placental mammals have been mapped for their entire genome. Okay, and around 2006, at least 2 of the marsupials have also been mapped for the genome. Okay, and when you go down on the evolutionary tree, the first version of complete genome is already mapped for platypus in 2007. But eventually, in order to compare different genome, we have one roadmap, and that's our evolutionary tree. And our evolutionary tree, in a sense, is also our legacy, and that's part of the reason we are interested in building this evolutionary tree, but not just with the molecules, but also with fossils too. Okay, let me revisit a age-old issue about the divergence time of modern mammal groups. Okay, this is the best study that had most extensive sampling. I know you cannot read the details, and in this DAISY dial there are 400 different families. But what's important is to bear in your mind, and this black line is geological time scale. This is the present time. This is the Middle Jurassic. This red circle represents the Cretaceous, and the Paleogene boundary, that's the 65 million year event when dinosaur got wiped out. When you map all this evolutionary lineage according to this study, and you have many groups that goes before the end of the Cretaceous. Okay, and here is the study by the NSF Tree of Life Mammal Project, the molecular team, they have sampled largest concatenated molecular data set, and many of the groups would go back into the Cretaceous by this molecular study. Of course, the morphological evidence do not always support such a claim, with exception of one case, and that's the split of marsupials and placentals. Okay. The molecular estimate of marsupial placental split goes roughly in the middle Jurassic, roughly from 140 to about 180 million years, okay? And no matter which study you're looking at, it's roughly in this time window, okay? The discovery of Jurassic, okay, definitely plays very closely with this molecular time estimate. However, for the rest of the late Cretaceous fossil, we still have a tremendous gap between the morphological estimate on major groups than the molecular time estimates, okay? Okay, and here is one of the early fossils called Eomaya. This is not from Jurassic, this is from early Cretaceous, okay? And this is Sinodelphis, and this is putatively related to the marsupial clade, okay? At least I think so, okay? Now, when you map this fossil record onto the time scale and following evolutionary genealogical tree, about 30 years ago, we largely working with the fossils from this time interval, okay? About 10 years ago, we start to have Eomaya, Sinodelphis, and we mapped the fossil record to this level. And in the more recent time, we can definitely push the split between placental on one side, marsupial on the other, down to this time interval, which fall comfortably within the molecular estimate of this group's divergence, okay? So it's good to have concordance, but we do not always have concordance in the sense that we still have this unresolvable issue about when the modern placental groups, not all mammals, just placental group, start to appear on Earth, okay? And with this kind of fossil, we can also answer some ecological functional questions in a limited way. And here is a reconstruction of Sinodelphis. Let's focus on the wrist, okay? And by looking at the wrist, we can tell that in the form closely related to marsupials, there are certain bones that are far more robust than the form that are closely related to placentals, okay? And it's also very interesting, the shape of the claw and the proportion of the fingers can also give us an inference about how this animal actually moved around, okay? Well, let's back step a little bit to follow this line of discussion, okay? And here is the modern opossum groups, and these are largely South American extant opossums, okay? And we have woody opossum that is arboreal, and we have mouse opossum that is also arboreal, but we have gray opossum that is terrestrial, and so like our backyard Virginia opossum is a largely terrestrial, but it's also capable of climbing. And among the terrestrial opossums, we can have swimmers, okay? Now, it turns out all this habitat preference or niche specialization are also represented by the finger proportions, okay? How so? Okay. So let's say here is Metacaris. Okay, it's one of the possums, and it's definitely known as ground living. And here is our familiar Virginia opossum, Didelphis, is a largely ground living, but it's also capable of climbing. Okay, and these opossums are full-time tree living. Okay, so in the specialized form that stay more time on the tree because they have to grab and hold, the finger proportions are different. Essentially, the farther end of the finger tend to be longer, okay? And same pattern can be also detected in modern placental mammals. If you map the earliest marsupials and the earliest placental from China, their finger proportion definitely falls onto the climber side than on the terrestrial side. All right, so we get some level of inference about how they moved around, okay? Now here is a bit more complicated, okay? Again, we're focusing on the hand bones called metacarpals, and we're focusing on the finger bones called proximal phalanx and the intermediate phalanx. So you map this bone along this axis, you map this bone on this axis, and then you map this bone on this axis. Essentially, this plot shows you how by finger proportion you can allocate the habitat preference of this modern primates and the marsupials in their distribution, okay? So that's how we understand we can use the finger proportion to distinguish different habitat preference. Let's bring the fossils into this discussion, okay? And these triconodons and multituberculates, these are extinct fossil groups, okay? These lived from Jurassic to Cretaceous. The majority of this, so far as we know, they're more like terrestrial mammals, okay? But by the time we get to a near mammal relative called Symmetrodon, they are also more likely, or the majority known so far, are also terrestrial, okay? But by the time we get to the modern placental relatives, marsupial relative, they are definitely closer to the tree-living primates and tree-living marsupials. So associated with the rise of our modern group, that is placental on one side, marsupial on the other, there is definitely a shift on locomotory adaptation and habitat preference. Essentially, somewhere around the rise of modern mammal group of Therian, we got into the tree, or we had a better capacity to get into the tree. Now this specialization is not isolated. We used to have the stereotype. Before the Cretaceous Paleogene extinction, somehow we have very limited diversity of modern mammals. And we also have a just so story to go with it because these early mammals lived in the time of dinosaur, share the same ecosystem with dinosaur, and suppressed by the dinosaur, they just fail to gain the great ecological possibility that they can realize until the dinosaurs are all gone, okay? So that's what we understand then, okay? But this is what we understand now, okay? Well, around 2000, 2006, we start to have semi-aquatic form discovered in the fossil, interpreted from some of the older fossil record, okay? And start from 2001, 2005, we get large enough of a mammal we know for fact can eat a dinosaur, but generally capable of carnivory. And that was considered unlikely before the recent time, okay? And in 2005, we discovered this form called a fruitifossor, and it has a very unusual specialization, otherwise only known in aardvarks and armadillos. That is, they have this peculiar teeth that's usually associated with tongue feeding of colonial insects, highly specialized, used to be considered as only possible with extant mammals, yet in a totally unrelated Mesozoic group, we found the same pattern, okay? Of course, we have now multiple forms that can climb the tree, and once you can climb the tree, it's only a jump away from gliding. So, okay, this is what we used to know, And this is what we now know for last 20 years of hard working by paleontologists going to all corners of the world, find the best fossil you can, okay? And also in the case of China, the great ingenuity of all these peasants try to do better for themselves, dug out all these great fossils. So what our Jurassic ancestor can tell us, okay? Well, we can tell that they're not just a bunch of Mesozoic roadkills, okay? They're flattened fossil, but they can definitely tell us about how the integumentary structure of mammals that characterize our whole group arise in the fossil record. And they can definitely tell us about the general growth pattern we can with confidence tie to the mammalian, the most fundamental mammalian adaptation, that's mammary glands and lactation, okay? And we can also tell the general pattern how in the earliest phase of the mammalian evolution, we start to have the larger brain, okay? And we can now also tell that no later than Middle Jurassic, we start to have the three main branch of extant mammal lineages, and that's placentals, marsupials, and the monotremes. And the rise of the therian mammals, so the placental and the marsupials, is definitely accompanied by some very interesting ecological diversification. It's more than just luck. It's really with good fingers, and we managed to hang on this next episode of evolutionary diversification in Jurassic. Thank you.